I say, amazing and much more than I would have anticipated. Well, it's just a real honor to be here at the National Academies building with colleagues from both Israel and the United States. And again, special thanks to the both the National Academy of Sciences and the Blavatnik Foundation. And again, thanks to our Israeli colleagues for making this long trip. And so we're very grateful that you can all be here. So I'm going to talk about germline and ancestral risks for lung cancer somatic mutations with a rather different topic that I'm throwing in on with some updates on genomic medicine for lung cancer. These are conflicts of interest, and of particular note, because I'm going to talk about it, is the licensing of patents for EGFR and ERBB2 inhibitors to Bayer. Just the outline, I'm first going to talk briefly about cancer in a general sense as a disease of the somatic genome, which will be familiar to you all, but I want to sort of introduce it. Second, the impact of genomically targeted therapies in lung cancer. Third, targeting EGFR and ERBB2 exon 20 insertion mutants. Fourth, somatic EGFR and KRAS mutations with respect to germline ancestry, the original topic. And then finally, summing up and discussion. And if there's a little bonus time, I'll throw in a couple slides on U2AF1 splice factor mutation functions in lung cancer. All right, so just in way of introduction, cancer genomics, somatic and germline. I think this is familiar to obviously many, but not all of you here. And so here's a little bit of a philosophical view, and we can go back to the Psalms, and in English translation it says, "For you formed my inward parts, you knit me together in my mother's womb. I am fearfully and wonderfully made." So if we make an analogy here, we can say the genome teaches us both about the wonderful, that is to say our healthy cells, and the fearful, our cancer cells. And our knowledge of the genome may help us turn fear into wonder. And I think that we are seeing the beginnings of this move from fear into wonder with what we can do already today in cancer diagnosis and detection and therapy, and more in what we will be able to do tomorrow. The key types of cancer genome alteration that we can see with genomic studies, and we've seen this from other speakers' talks, include mutations, copy number changes, chromosomal rearrangements, and infections. And I should say, I've been blessed to work with Gadi Getz for the last couple decades, and Gadi has really developed a lot of methods to figure out each one of these. And I've been riding on Gadi's coattails, and I've been riding on some other coattails too that I'll talk about. So cancer is a disease of the somatic genome. That is to say, the cancer mutations for the greatest part are present in the cancer DNA, but not in the germline DNA. And yet those risks of somatic mutation can be modulated by germline variation. This is important because the somatic alterations are internally controlled. So the comparison between the germline and the cancer defines the cancer-specific alterations and allows for accurate discovery and diagnosis. So importantly, these alterations provide a therapeutic window for treatment. So just like a bacterium or a virus has a different genome than we do, very different genome, and so we can make an antibacterial or antiviral treatment that doesn't affect our cells in principle. This is to a lesser extent, but significantly true of cancers, in that targeted therapies can kill cancer cells with unique genomic alterations. And immunotherapies, as we'll probably hear from Yardena momentarily, can kill cancer cells with unique neoantigens. And so here we've got a real distinction. So in the next slide, I said I've been riding on Gadi's coattails. So as Eric pointed out, we've been riding on the coattails of our colleagues in the sequencing instrumentation industry. So this is a slide that we'll say that Eric made this slide, which is approximately true, since it came from the NHGRI. It's as true as me saying that I made some of the upcoming findings that I'll discuss. So we've just seen, as Eric mentioned, the first human genome sequencing in the late 1990s, costing several $1 billion. Sequencing today of a genome, about $300, a 10 million-fold improvement. So I always like to say, imagine that housing prices had decreased by the same rate as the genome sequencing has improved, right? Like we would each have like our own personal Mar-a-Lago in every city, for example, just to put that in comparison. You know, I don't think any technology has improved at the rate that genomics has. I think we all know this, but every time one thinks about it, it's really quite miraculous. And it's thanks to our colleagues, starting with our colleagues at Solexa and others. So what's been the impact of targeted therapies in lung cancer in particular? And I think it's been quite dramatic, actually. So when I started at Dana-Farber, in 1998, the human genome was being sequenced at a cost of $3 billion. And I thought, well, I'm going to use genomics to study cancer. And how did I decide? I actually looked at this list from the American Cancer Society and said, oh, lung cancer's at the top of the list. It's important. And at that time, there were about 160,000 deaths per year from lung cancer in the US. If we looked at lung cancer treatments at this time, basically they were all the same and they all didn't work. And the median survival was around 8 months for metastatic lung cancer, whether you had adenocarcinoma, whether you had squamous cell carcinoma. This was a New England Journal paper. Oh, I see the bottom is sort of under a little black bar there, but New England Journal paper from 2002, and this was like the best we could do. And so it's wonderful to see we can do better. So in 2001, importance of international scientific meetings, Bill Sellers and this guy was me. Our genomes change, right? It makes a difference. I just want to say, right? This is. I don't know if this is a genomic change, an epigenomic change, or a transcriptomic change, but maybe some of each. Anyway, we were on a trip to Switzerland, and we were talking about the BCR-ABL inhibitors for chronic myeloid leukemia, and we were talking about the initial sequencing of the human genome, and what can we do? And Bill said, well, let's sequence the receptor tyrosine kinase genes, which was like ultra high throughput sequencing, right? We sequenced 30,000 bases in this case for lung cancer, 58 paired samples, and this was like, right? This was like way more than people had done, except for Victor, who was sequencing at bigger scale at that time, but more than anybody else had done. Right, and so in this study, right, we identified our first three mutations. It was L858R mutations in the epidermal growth factor receptor, or EGFR gene, all in non-smoking women with adenocarcinoma from Japan. And I'll come to the importance of this momentarily. But one thing that was super interesting to us, the BRAF kinase, this is the catalytic loop. This aspartic acid here is conserved in all kinase genes. There's a BRAF somatic mutation here that we had actually just recently identified, an L to R mutation in the same position in a serine 3 and 9 kinase gene, so we were super excited. And here are a few important colleagues in this work. And we found mutations in multiple domains, activation domain, the nucleotide binding loop, deletions in this part of the protein structure, insertions in this part of the protein structure. Guillermo Pérez and Jeff Lee led the sequencing, Titus Bogan and Mike Eck the structural comparison. So there was a real mystery at this time. There was a lung cancer kinase inhibitor, gefitinib, and this is a patient of Bruce Johnson's. You can just see total disappearance of cancer within 3 months of treatment. And David Carbone said, we see a complete Lazarus type of response that we've never seen before. There were 4 groups of patients with an improved response to gefitinib, those with adenocarcinoma, those from Japan, women, and non-smokers, exactly the characteristics of the 3 patients in whom we found the first mutations. And so, in fact, working with Pasiana and Bruce Johnson, we showed that patients who had responded to gefitinib had EGFR mutations in their cancers. Those who did not respond did not have EGFR mutations. And similar findings were made simultaneously, and I think this helped the impact a lot, by Tom Lynch and Daniel Haber at Memorial Sloan Kettering Cancer Center, and by William Pao and Harold Varmus at, first one's at Massachusetts General Hospital, and by Will Pao and Harold Varmus at Memorial Sloan Kettering. So this led to the development of new EGFR inhibitors like osimertinib. And, okay, I'm just going to flip back a second. Okay, this was where this was where cancer clinical trials were, okay, and this is where cancer clinical trials are, right? This is survival over time, progression-free survival. This is time. This is placebo. This is the EGFR inhibitor osimertinib, right? Huge improvement, a hazard ratio of 0.16, meaning 16% chance of getting progression compared to the original. Huge, huge, huge improvements. So there are a lot of receptor tyrosine kinase gene alterations in lung adenocarcinoma. Many, many genes in this pathway are altered. There's even more than this. And since, right, this is all the lung cancer drugs approved before the discovery of EGFR mutations in 2004. And this is what's been approved since. So like all history prior to 2004 versus 2025, about the same. A remarkable level of progress. And we see, right, so the US population has increased by over 25% since 1998, right? People have gotten older, but the lung cancer death rate has decreased by more than 20%. It's partly due to smoking cessation, it's partly due to early detection, it's partly due to immunotherapy, and it's partly due to targeted therapy. So it's a big decrease in the death rate, and this is true for other cancers as well, but this is a dramatic one, and I think it's in part due to these findings. Okay, so I'm going to talk about something brand new, a new example of a genomic medicine for cancer, an EGFR and ERBB2 exon 20 insertion mutant inhibitor, and this has been led by Heidi Grulich in my group, and we've been collaborating with Bayer on this project for a long time. So shortly after our mutation discovery, right, we saw, okay, many of the major EGFR mutations are highly sensitive to gefitinib or erlotinib, as shown here in a soft agar assay, which is a measurement of oncogenic transformation. You can see here these are NIH3D3 cells transformed with the wild type or each of these mutants. The wild type is not inhibited very well. These three classes of mutants inhibited very well, but these exon 20 insertion mutants inhibited no better than the wild type. And in fact, patients with these mutations do not benefit from EGFR inhibitors compared to patients with other mutations, shown here in black in EGFR work of John Hamack and colleagues at the MD Anderson Cancer Center. Okay, around a little bit after we reported EGFR mutations, I should mention Phil Stevens and Peter Campbell, Andy Futrell, Mike Stratton and colleagues reported kinase domain mutations in ERBB2, which are very similar to those drug-insensitive EGFR kinase mutations that I just showed. So for the last 20 years, people have been struggling to develop EGFR and ERBB2 insertion mutant inhibitors, and so we've been working on this, a team, Heidi Grulich, together with Franzi Siegel and Stefan Siegel at Bayer. Franzi and Stefan, despite having the same name, they are neither a married couple nor siblings, no relation between them. But this collaboration actually started at an international meeting, the AACR, American and Indian Cancer Associations meeting, so I think a lot of promise for what can come out of a binational meeting. So hopefully something as dramatic or more will come out of this meeting. And the compound cevabertinib, which is now in phase 3 clinical trials, this is showing data for EGFR inhibition. It's about 40-fold inhibition of the EGFR exon 20 insertion mutants compared to wild type. Okay, molecular structure, this was actually developed out of a serine 3-adenine kinase screen. Quite coincidentally, it happened to specifically inhibit this and some other EGFR mutants. A structure completely different from other previous EGFR inhibitors, and this is how it looks. This is activity here against, these are BAF3 cells transformed with, BAF3 cells are IL-3 dependent. If you don't have IL-3, you can transform them with a variety of kinases, and you can see ERBB2 inhibition compared to the parental cells, many hundreds or 1,000-fold difference. Again, wild-type EGFR would probably look like this. Wild-type ERBB2, there's no selectivity for these inhibitors compared to wild-type ERBB2 inhibition. This is a patient-derived xenograft, and you can see here significant tumor shrinkage of these xenografts grown in mice compared to the wild type. And now on the next slide is a design of the clinical trials, and there are three major cohorts shown here. Those that have not had previous ERBB2 therapies, those that have been treated with ERBB2 antibody drug conjugates, and those that have had not any systemic therapy, and has shown benefit in all three of these cohorts. And this is actually one of the first treatment patients with ERBB2 mutant lung cancer treated with cevabertinib. And you can see here there's a transverse CT section. You can see here shrinking, shrinking, shrinking. Well, not shrinking anymore, but you see significant tumor shrinkage early on. And what does this mean for patients? So, all right, so this is a type of plot that you would never see 25 years ago. It's called a waterfall plot. If you're going above the x-axis, the cancer is growing. If you're going below the x-axis, the cancer is shrinking. And as I say, 25 years ago, I think you would have had to have a mountain plot, if you will, with all the cancers growing. And today, I'd say for cancer therapeutic trials, seeing a plot like this is now routine. And what we've seen here, this is blinded, independent of the investigators' analysis. And what you can see here, if you look at a 30% shrinkage cutoff, you see about 70% of these patients are exhibiting at least 30% of shrinkage. And it's now currently in phase 3 clinical trials. So this is a pretty remarkable analysis of a new compound based on genomic medicine. And this manuscript came out in the New England Journal of Medicine on Friday of last week, so I really thought I would share it. So just a summary for this, a new non-covalent inhibitor of EGFR and ERBB2, very high selectivity for mutant receptor tyrosine kinases relative to wild-type EGFR, including the hitherto undruggable exon 20 insertion mutants, very encouraging results in phase 1 and 2 clinical trials, and a chemotherapy-controlled trial is now underway. So now my title topic, somatic mutation and germline risk, work of Jen Karajan in close collaboration with Sasha Gusev at Dana-Farber, and our clinical collaborators Oscar Arieta and Andres Cardona. So I mentioned that we found there was a higher rate of response to erlotinib and gefitinib in Japan than in the US, and it turns out, in fact, in our initial study and beyond, the somatic mutation frequency for EGFR is about 10% in lung cancers from people of European or African descent, but it's about 50% in people from East Asian descent. These mutations are most common in patients who are non-smokers. However, there's no evidence that smoking is protective against development of cancer with these mutations. It's just that lung cancers with other mutations, especially KRAS mutations, develop more in smokers. So is this a germline difference or environmental? So if we look by country, and this was work of our colleagues, and this is our paper from 2021, we can see here, East Asia, it's about 50% mutation frequency in EGFR, about 10%, again, in Europe or the US, people of African or European descent. And in Latin America, well, it's about 50% in Peru, mostly people of Native American ancestry. It's about 10% in Argentina, mostly people of European ancestry. And in Brazil, Colombia, and Mexico, where people have a mix of European and Native American ancestry, the relative proportion of EGFR mutations in lung cancers in between that for people of European ancestry and those of Asian ancestry. And of course, the Native Americans are believed to have migrated to the Americas from Asia via an ice bridge across the Bering Strait between Siberia and Alaska, potentially bringing with them a risk allele or risk alleles. So we wanted to test that, and we looked at 2 cohorts of patients from our colleagues in Mexico and Colombia. These are DNA samples isolated from lung cancer in the course of clinical care, and the patients were consented for germline as well as somatic genomic studies. So we looked at a panel of several hundred cancer genes, and we then called the genotype of the germline single nucleotide polymorphisms using both on-target and off-target reads, and then did genome-wide imputation of SNPs throughout the genome. And then we measured global ancestry based on principal component analysis of the germline SNPs. And so what you can see here is that as you have an increased fraction of Native American ancestry, you have an increased probability of EGFR mutation rate and a decreased probability of KRAS mutations. And this is true both within the Mexican population and within the Colombian population. And in addition, we looked at local ancestry and found that correlated better with each of these somatic mutations than did global ancestry, again, consistent with a germline rather than an environmental factor. So this is just another analysis in non-smokers only, saying that it wasn't smoking confounded. We see no effect of smoking signature or of age on these mutations, but we see Native American ancestry is positively correlated with EGFR mutation and negatively correlated with KRAS mutation. So our conclusion here is that somatic EGFR mutation is positively correlated with Native American ancestry. Somatic KRAS mutation is negatively correlated with Native American ancestry. Our unproven hypothesis is that one or more alleles that are common to people of both East Asian and Native American ancestry increases the risk for lung cancer with somatic EGFR mutations and decreases the risk for lung cancer with somatic KRAS mutations. So we're still looking. Our goal is we are hoping to collect a larger cohort of lung cancer and blood specimens from a very large cohort of Latin American patients. We're also interested in building on large existing cancer genome data collections, and we're just about to start examining the Foundation Medicine collection for this purpose. We're trying to get other large collections as well, and we're applying new reference panels which have better references for genomes of diverse ancestries, like this short read panel from Koenig, and potentially new long read panels that are coming up. I'm going to skip this and just say, I'm just going to give a quick peek here, okay? This is David Walter. I'm 2 minutes ahead here. All right, he's been working on U2AF1 mutants, which are a splicing factor. Back in 2012, this paper by Marcin and Malinsky, we identified a number of splice factor mutations associated with lung adenocarcinoma, U2AF1 mutations and RBM10 mutations, notable among them, and I think we'll hear new work on RBM10 tomorrow from Nabia Ayoud, and so looking forward to hearing about that. And super briefly, he did, it's a really cool thing. S34F is the repetitive mutation of U2AF1. David did prime editing. There's exon skipping of one of the mutant forms of KRAS, reverted by the S34F, and back reverted by the wild type. And so it looks like some of these splice factor mutations may be a secondary mutation that's existing to correct a splicing alteration caused by the genomic impact of oncogenic mutations in cancer. So EGFR mutation in ancestry, we're looking for a locus, and I welcome your advice in the panel or afterwards. I've mentioned cevabertinib that's in clinical trials, and these results of U2AF1 mutation that may modulate KRAS mutation-induced alternative splicing. So thanking a number of people, Jen Karajang and Anders Dolman, for the work on EGFR mutation and ancestry, EGFR and ERB2 inhibitors, Heidi Grulich, U2AF1 mutations and splicing modulations, David Walter, and again, as I say, I'm particularly indebted over the course of my career for a long-standing collaboration with Gadi Getz, who's taught me a lot over these years. So thank you for your attention. I can take any clarifying questions, and then we will move on to the last talk of the session from Yardena.